Good afternoon, everyone. We are the members of Group 2 and we will be doing lysosomes. Our group members include Ricky Samwaru, Tanya Ali Abudu, Davina Emily Prasad, Emily Grace Shadunat. I will now hand you over to Tanya.
What are lysosomes? Lysosomes are eukaryotic membrane-bound cells organelles that contain digestive enzymes. This means that it has an acidic inferior. The enzymes it contains are called hydrolytic enzymes. They break down food from molecules, especially proteins and other complex molecules. I will now hand you over to Ms. Divina.
Structure of the lysosomes. The first point says that they are formed by budding off of the Golgi apparatus. Now the hydrolytic enzymes within them are formed in the endoplasmic reticulum. The enzymes are attached with a molecule called mannose-6-phosphate, which is transported to the Golgi apparatus in viscose and then packaged into the lysosomes. There are many different types of lysosomes, including protease, which breaks down proteins, amylase, which breaks down starches into sugars, nuclease, which breaks down nucleic acid, and so on. The second point, they are generally very small and spherical in shape. As you can see depicted in the picture at the side, it is spherical in shape, but this is a magnified version of the lysosome. That's why you can see it big. And the third point, they are made up of a lipid bilayer. Now, bilayer means two layers. So this layer seals off the fluid that has like 40 variety of hydrolytic enzymes, and they're made up of phospholipids. Now, phospholipids have hydrophilic phosphate group heads, a glycerol molecule, and a hydrophobic fatty acid tail. And if this does not seal it off, this would spill into the cell and destroy the whole cell. That's why it has to be protected from the rest of the cell. And the last point, the membrane contains highly glycosylated lysosomal associated membrane proteins known as LAMP and lysosomal integral membrane proteins known as LIMP. Now, the LIMPs and the LAMPs form a coat on the inner surface of the membrane. The membrane is also containing hydrogen proton pumps that would help to maintain the pH conditions in the lysosome. So since the inside of the lysosome is acidic, it can be maintained by the proton pump, which would is inside the lumen. The lumen is the inner part that is enclosed with the lysosomal membrane. And the hydrogen ions will make sure that the lysosomal enzyme functions properly. I'll now hand you over to Tanya and Emily.
Functions of lysosomes. The first function is that they break down large molecules into small molecules. This is possible through the process of hydrolysis. Lysosomes can be thought of as the digestive system of the cell because it breaks down molecules using enzymes. By doing this, they provide essential nutrients for the rest of the cell.
The second function is they ingest bacteria to fight against infection. Lysosomes fuses with an endocytic vesicle. An endocytic vesicle is a vesicle that brings particles into the cell. When bacteria enters the body, the white blood cells called the phagocyte, which is a vesicle, ingest the bacteria to break it down. The lysosomes fuse with the vesicle, then breaks down the bacteria. This process is called phagocytosis.
The third function is that lysosomes aid in the removal of dead cells in tissues. Examples of such cells are cells in the outer layer of the skin and mucous membrane linings of the body. If the cell is damaged beyond repair, lysosomes can help it to self-destruct in a process called programmed self-death. I'll now hand you over to Tanya.
In conclusion, the lysosomes aid in the digestion, removal of waste material in the cells, and it helps to process energy. When we absorb food, the lysosomes break down macromolecules into unstable energy. It is quite similar to the starvation response. When your body does not have enough food accessible, it digests organelles inside of the cell to obtain nutrients. We are positive that our listeners this afternoon has gained much information on the structures and function of lysosomes. This concludes our presentation. Thank you for listening.